Luca Comai. Let's continue with the 10 most important features of meiosis. Number 5. Meiosis consists of 2 cell divisions. What happens in these divisions? This is a simplified timeline of the major events. Meiosis 1 represents the key cell division, while meiosis 2 is just like mitosis. In meiosis 1, chromosomes pair, recombine, and the 2 homologs go to opposite poles. Number 6. One way to recapitulate meiosis is to examine what happens to chromosome numbers and to DNA content per cell. Starting with DNA content at 2 copies per cell, we see it doubling during the S phase. In G2, there are actually 4 DNA copies of any gene. The amount of DNA is reduced at each division until a single copy is placed in the gamete. Chromosome number follows a different path. Contrary to what one may think, the chromosome number stays the same during S phase and G2. The chromosome number is halved at the end of anaphase 1 when the 2 cells produced by meiosis 1 inherit half the chromosome number of the G1 cell. It is easy to be confused by the fact that each chromosome has 2 chromatids. The next division keeps the same chromosome number and for that reason is comparable to mitosis. Number 7. Missegregation of chromosomes leads to aneuploidy, the condition of having 1 or more extra or missing chromosomes than the regular number. This results in an uneven chromosome set. To clarify this, let's consider the karyotype of a diploid cell with a total number of 4 chromosomes. X, the basal chromosome number, in this case is 2. This is also the number of chromosomes expected in a gamete of this species, called N. This aneuploid has 5 chromosomes instead of 4, 3 copies of chromosome 1 and 2 copies of chromosome 2. In this karyotype, chromosome 1 is trisomic and the condition is referred to as trisomy 1. In a second example, a cell that should be diploid displays 3 chromosomes instead of 4. There is a single copy of chromosome 2 and the condition is called monosomy 2. Why is aneuploidy bad? Each species has evolved to have an appropriate balance of all proteins. Trisomy of chromosome 2, for example, means that the proteins encoded by the genes on chromosome 2 will be, in average, produced with a concentration value of 3, while the rest of the proteins are produced at the expected level of 2. The loss of balance for certain proteins has severe consequences on function of the cell. Number 8. What causes aneuploidy? At anaphase, the 2 chromosomes of a pair during anaphase 1 and the 2 chromatids of a chromosome during anaphase 2 disjoin, that is, they separate. Failure to separate is called nondisjunction. Let's see what that looks like. At anaphase 1, we expect to see 2 replicated chromosomes move apart from each other. Instead, during nondisjunction, they somewhat stick together and go to the same pole. At anaphase 2, A2, we expect to see the 2 chromatids of a chromosome separate and go to opposite poles, but instead, they stick together and wind up in a single daughter cell. Consider the result of nondisjunction at A1. 2 of the 4 cell products, in this case the gametes, inherit 2 copies of the missegregated chromosomes. The other 2 cells inherit none. For clarity, we only show the events affecting a single chromosome type. The other chromosomes segregate properly and are not shown. Consider now the results of nondisjunction at anaphase 2, A2. 2 of the 4 meiotic products will be normal. The other 2 will not. One will inherit both chromatids, which once separated will result in 2 chromosomes in this particular cell, while the sister cell will not inherit any copy. In summary, the earlier non-disjunction occurs, the more pervasive the outcome. We now arrive to meiosis facts number 9 and 10. What about them? Well, I have a confession to make. I'm running out of facts and I'm getting tired. So let's end here. Thank you.